Today I'm going to talk about visuospatial dysfunction in aging and dementia. And I'm going to emphasize the dementia part more than aging, as you'll see. But a lot of this will relate to normal aging as well. And as I'm describing some of the deficits that we see related to dementia, I'd like you to think about how it might impact our responses or the responses of people with dementia to the built environment. And also maybe think about how you would build an environment knowing the kinds of deficits people might have. So when we talk about visuospatial dysfunction, we are really talking about a number of different aspects of visual deficits in visual cognition. They can be broadly divided into those related to objects and those related to space. And I've just listed a few things here. There are, of course, deficits in visual perception, so you can have visual object agnosia, not knowing what an object is, or you can have a deficit in analyzing and synthesizing components into a whole object. In terms of spatial deficits, you can have difficulty localizing points in space, judging direction and distance, or topographical orientation. And some aspects of cognition actually involve both, things like visuoconstructive, defective assembling performance. And that can be either in three-dimensional space or even in two-dimensional if you're drawing complex objects, for example. And that distinction between object and space, this is a very simplistic view I'm going to present here. It's much more sophisticated. But the cortex honors this distinction between spatial and object processing to some degree. There are two distinct streams of cortical activity. The dorsal stream going from occipital cortex into parietal cortex regions is involved largely in spatial encoding, and the ventral stream from occipital cortex into temporal lobe cortex is involved with encoding properties of objects. Again, that's very simplistic, but good for this purpose. So the disorders that I'm going to be talking about, I'm going to talk about two neurodegenerative diseases, Alzheimer's disease and dementia with Lewy bodies. And Alzheimer's disease, many of you are probably familiar with Alzheimer's disease. It's a neurodegenerative disease that produces atrophy and loss of neurons, loss of synapses in the cortex and in these medial temporal lobe structures, including the hippocampus and entorhinal cortex that Dr. Leutgeb just talked about. The primary pathologic hallmark of Alzheimer's disease are these microscopic inclusion bodies or lesions known as plaques and tangles. And the plaques and tangles and the atrophy and neuron loss are distributed in the mediotemporal lobe region and in the cortex, but not all aspects of cortex are affected. This is the brain of someone who had Alzheimer's disease, and if you look at the atrophy, you can see the temporal lobe region and the parietal lobe region are two cortical regions that are highly affected by the disease. So both of those correspond to the dorsal and ventral visual streams. So what we see in Alzheimer's disease clinically, as you probably know, the primary presenting complaint in Alzheimer's disease is memory impairment, but there are also deficits in other cognitive abilities, aphasia, a language deficit, and as we'll see, object knowledge deficits and deficits in visuospatial abilities. Okay, turning to dementia with Lewy bodies, it's similar to Alzheimer's disease, and in fact, most cases of dementia with Lewy bodies have the plaque and tangle pathology in addition to a different pathology. The different pathology is usually associated with Parkinson's disease. And in Parkinson's disease, we see atrophy in cortical regions, but also in subcortical regions. And the primary histopathologic lesion or inclusion in Parkinson's disease is the Lewy body. And these Lewy bodies in Parkinson's disease are found in subcortical structures like the substantia nigra, and that leads to the movement problems that you often see, the tremor and gait problems and so on. But in dementia with Lewy bodies, these Lewy bodies are also found in the cortex. And they're found in some of the same areas where we see Alzheimer pathology. So the temporal cortex, the parietal cortex, and so on. So it produces a progressive dementia similar to Alzheimer's disease, but there are some mild Parkinson movement disorder. Also, visual hallucinations are prominent in dementia with Lewy bodies, and they have fluctuation in attention or consciousness. And one thing interesting in the comparison of dementia with Lewy bodies to Alzheimer's disease is that patients with Lewy bodies tend to have a greater amount of hypometabolism in these posterior brain regions involved in vision and visual processing. So this is just a PET scan showing hypometabolism of glucose. A normal person, when it's red, that means that area is lighting up. It's using a lot of glucose. We see that's reduced in this person with Alzheimer's disease in these posterior regions, and even more reduced in patients with dementia with Lewy bodies. So we might expect them to have worse visuospatial deficits than patients with Alzheimer's disease. So now I'm going to just go through a couple of studies that we've done comparing these groups on these aspects of visual cognition, starting with visual object. A study we did, we asked the question, is object recognition and synthesis impaired in patients with pure Alzheimer's disease or in those with dementia with Lewy bodies? And to look at this, we used a task called the Hooper Visual Organization Test. And in this test, you're shown 25 objects, but the objects are divided up, kind of sectioned and randomly placed. And you have to look at it, synthesize the parts in order to identify the whole. And we asked the person just to name the whole object. Well, as I mentioned, patients with dementia, Alzheimer's disease in particular, sometimes have a language problem. And it could be they just couldn't get it because they couldn't name the object. So after we were done with the test, we showed them each object in its entirety and asked them just to name it so we could correct for that problem. So this is just an example. I think you'd see a stimulus like this and you'd have to say what it is. Okay, it's a chair. You'd see this. So hopefully everybody knows what this is. If you don't, please see me after the session. That's an airplane. Okay. So we presented the test, and here are our findings. Compared to normal control subjects, both dementia with Lewy bodies and Alzheimer's disease patients were impaired, so they had difficulty on this task. The dementia with Lewy body patients were slightly but significantly worse than patients with Alzheimer's disease. And the same pattern held if we corrected for the inability to name objects. So definitely these dementias cause a difficulty in integrating components into a single visual object. One interesting aspect we decided to look at, because dementia with Lewy bodies also often has concomitant Alzheimer's disease pathology, we split them up into those with low or high levels of Alzheimer pathology. You can see that had no impact, so it really looked like it was the Lewy body pathology that was driving their worst performance than patients with pure Alzheimer's disease. Another aspect of visual cognition that we look for deficits in are visual constructive abilities. As I mentioned, drawing is one way to do that. We use a very simple test, a clock drawing test. Shown here. There are two components, two ways we do this. One is, we call it the command condition. That's where I'd give you a blank sheet of paper, and I'd say, draw a clock, put in all the numbers, set the hands at 10 past 11, and then you just perform the task. So you can see that assesses visual constructive ability. You have to draw and construct it, but also conceptual ability or conceptual knowledge. You have to understand what a clock is, what are the components of a clock, how do they relate to each other. To try to separate those two, we also give a copy condition where I just give you that picture I showed you before of a clock and say, copy this in the space below it. That way I can look to see if you have a visual constructive problem independently of having to really know the conceptual aspects of a clock. You don't even have to know what a clock is in order to copy the figure that you're looking And what we see when we look at patients with Alzheimer's disease, they're clearly impaired compared to control subjects on clock drawing command performance. But they're also impaired even in the copy condition, but they're worse in command than copy, which implies there may be this additional conceptual deficit on top of a visual constructive problem on this task. One way we can look at that is to look at the kinds of errors that are made. Take a qualitative look at it. So we can look for spatial errors, various kinds of spatial errors. If a person is constructing and has a spatial deficit, they may make these kinds of errors. This patient neglected the left half of space, you can see. This person had planning difficulties where they would leave gaps between numbers. They knew the anchor points should be here and here, but their planning was off, so they left gaps. Same thing here. Here you can see they knew a clock had numbers, but the spatial relationship between the numbers and the boundary of the clock were violated. These are not so much spatial, but these are perseverative errors, where a person starts to draw hands and just keeps drawing more and more, or this, where they put in the numbers and just keep going around again and again. So, and that's the timer I'm using for this talk, so I might take a little more time. You can also make conceptual errors, right? So even if your construction is right, if you don't understand what a clock is or start to lose your knowledge of it, then you might show errors like this. This, interestingly enough, is a stimulus-bound error, is probably related to damage to the frontal lobes. And we think what the person is drawn to the 10 and 11 because the instructions said set the hands at 10 past 11. If you can't represent that time, then you might be drawn to the 10 and 11. This person made a conceptual error. The arrows are going the wrong direction. This person clearly had knew a clock had numbers, but definitely had a conceptual deficit here, and told us that he was drawing a digital clock. So, and then you can see other aspects of these kinds of errors. So by doing that error analysis, we can look to see, is there a difference in the constructional versus the conceptual? And we think there is. Also, there's a progression in these deficits in clock drawing over time. Over a 2-year period, you can see command condition is worse than copy in patients with Alzheimer's disease, but both decline over a 2-year period. And here's an individual patient just showing their decline. The first time we saw this patient, here was their command clock. They clearly had a conceptual deficit in what a clock represented, but their visual constructive ability was actually good. They could copy the model of the clock without much difficulty. A year later, the conceptual aspect had clearly declined. The constructive aspect, not so much. They still did a pretty good job. There was a few errors you can see in setting the hands at 10 past 11. By the 3rd year of this progressive dementia, you can see they had completely lost what a clock was. We haven't done a large study in dementia with Lewy bodies, but this is a look that we took. Each column here represents one dementia with Lewy body patient and one Alzheimer patient who were matched in terms of their global level of cognitive ability. So their memory deficit was about the same, language was about the same, and so on. But you can see, and that's true for each column, they're matched that way. And in general, what we see is that patients with dementia with Lewy bodies have a worse visual constructive deficit than patients with Alzheimer's disease at the same level of dementia. Okay, another aspect of visual cognition I wanted to mention is memory for spatial locations, a study that we've done. We used something called the recognition span task, and in this task, a person must remember an increasing number of stimuli across trials. There's a 15-second stimulus presentation, 10-second delay, and we can assess working memory for spatial and verbal information. And I'll show you just an example. Person is shown this. Say, remember the location of that dot. Goes away. Which dot is the new dot? So all they have to do is point to the new one. So that one is in the same location. That's a new one. Okay, those go away. Point to the new dot, goes away. Point to the new dot, and so on. So you continue that out until the person makes an error, and that's considered their memory span. And we do that twice and average it. And what we see is that both patients with Alzheimer's disease and patients with dementia with Lewy bodies are impaired on that task, even though it's a very simple task. It was actually developed for monkeys, non-human primates, and we adapted it. And you can also see that patients with dementia with Lewy bodies are significantly worse than patients with Alzheimer's disease on this task. So they have a real difficulty with working memory for spatial locations. But we were concerned, is it really visual or is it a general working memory problem? And it may be to some extent, because if you do a verbal analog, you see the same pattern of results. So it may not be specific to spatial information, but a general working memory deficit. Okay, another thing I wanted to talk about was integrating information in the visual system. And does Alzheimer's disease affect the ability to integrate information across those two visual streams I mentioned? So we're going to look at integration of information across the dorsal and ventral stream. To do this, we used a global motion coherence paradigm. And the person is seeing this. These are moving dots in this circle. And you have to discriminate the direction of coherent signal dots that are moving together in the same direction amid random noise. So you can see all of these with the dark arrows are going the same direction. The rest are random. Can you see the general flow of the dots to either the left or the right? So it's simple for a patient. They just have to say left or right, or they can even point. They don't even have to say. Then you vary the percentage of dots that are moving coherently systematically in order to get a motion discrimination threshold. This is the task we used. To look within the visual streams, we looked at segmentation of the coherently moving dots using luminance. So that's processed both space and luminance in the dorsal stream. We had a baseline condition where you just look for movement. You see there's a general flow in one direction. Then uncoupled, where the luminance doesn't help you. It's kind of random. And then, but the stimulus configuration is the same as the coupled condition, where the luminance is actually a cue to help you segment out the coherently moving dots, like that. We also did it using color and motion. And the reason we did that is they're processed in different streams largely. So the motion in the dorsal stream, color in the ventral stream, but it's the same idea. And I should mention, we borrowed these tasks from Dr. Albright here at the Salk. So again, we had a baseline, uncoupled condition, and the coupled condition. Okay? And basically what we saw is that patients with Alzheimer's disease showed the same benefit from having the cues segmented by luminance as normal control subjects, but they were impaired compared to control subjects, had a higher threshold when they had to integrate across the visual streams the color and the motion information. You can look at an index for that. We calculated that just to show the deficit in a clearer way. And you can see luminance motion within stream integration was normal in patients with Alzheimer's disease. Color motion or cross-stream integration was impaired compared to controls. When we looked at patients with dementia with Lewy bodies, we couldn't even get that far. They had a primary deficit in just seeing motion or discriminating the direction of motion, even in the baseline condition. So we saw that here compared to both normal control subjects and patients with Alzheimer's disease. And in the motion luminance condition that Alzheimer patients could do normally, dementia with Lewy body patients were severely impaired. So they have a strong deficit in visual integration. Okay, so this is the last study I wanted to show. So since my time's up, this is good. And this is, we wanted to look at visual search in patients with dementia, and patients with Alzheimer's disease, and those with dementia with Lewy bodies. And we used two visual search tasks. something called a single feature search, where a single feature distinguishes the target stimulus from the background noise. And you can see the person is simply looking for the presence or absence of a black dot. So in this case, the target is present. In this case, it's not. Usually when there's a single feature, there's something called the pop-out effect. And I'll explain that in just a second. And then the second task was a feature conjunction condition, where the target stimulus could vary from the distractors in two different ways. So it could be either luminance or color, or it could be shape. So again, you're just looking for a black dot, right? But now the distractors are different, so you have to integrate both the luminance or color information and the shape information in order to find the target. So it's a little more difficult. And this is what we typically see if you just measure reaction time to find the target. As you increase the number of distractors in the single feature search, usually there's no increase in reaction time because the stimulus just seems to pop out. So it doesn't matter how many different distractors there are, the target pops out at you. In contrast, in the feature conjunction search, there is a cost, so that the more distractors there are, the slower you are at finding the target. It's almost as if you're doing an item-by-item serial search until you can find the target. So we wanted to look at those two aspects. Here's just an example. This is the single feature. Just see if the black dot is there. This is the target present. This is target absent. And then the feature conjunction, you look for the black dot. There's target present. And hopefully everybody found it. And then target absent. And these were the results that we found. We were a little bit surprised, particularly in the patients with dementia with Lewy bodies that are shown here. Patients with Alzheimer's disease here. These are controls, patients with Parkinson's disease without dementia, and some patients with Parkinson's disease with dementia. But what we see is that DLB and Alzheimer's disease patients are slower overall than the rest to find the target, but they don't show any increase as the number of distractors increase. So they are showing the pop-out effect. So despite their other visuospatial deficits, patients with dementia with Lewy bodies show a normal pop-out effect. And some people think that the pop-out effect is really mediated by these very primary visual areas, even V1 or V2, in the occipital cortex, and those might be areas that are preserved still in dementia with Lewy bodies. In contrast, when we look at feature search, patients with dementia with Lewy bodies and those with Alzheimer's disease actually show a steeper slope than normal control subjects, so that they show a greater cost as the number of distractors increases. It's difficult to see, but if you take the difference between the 3-set distractor size or 12-set distractor size, you can see a little better. That's done here. So you can see that difference is much greater in DLB or dementia with Lewy bodies and Alzheimer patients compared to normal controls or the other Parkinson groups. So it looks like Alzheimer's disease and dementia with Lewy bodies cause inefficient serial search in the conjunction conditions, perhaps due to deficits in feature binding. So just to summarize, what we see in patients with dementia, particularly Alzheimer's disease and dementia with Lewy bodies, are deficits in object identification, particularly if you have to synthesize the features. Deficits in visual construction that you can see on drawings, or I didn't show you, but three-dimensional objects, building three-dimensional objects. They show deficits in spatial working memory that could be related to hippocampal dysfunction as well as their other visual deficits. And they have deficits in identifying the direction of motion, particularly patients with dementia with Lewy bodies, and deficits in integrating color and motion information. And finally, they do have some deficit in visual search, particularly if they have to integrate features of the visual stimulus. So finally, what are some possible implications for the built environment that a person with dementia has to live in? And I thought of a few things here, but would leave it to the experts to think about this more. So if you have difficulty, you might have difficulty identifying objects, particularly if they're in an unusual perspective or an unusual shape. So you can imagine, You know, if you're designing a doorknob in a strange way, a person with dementia might not recognize that as a doorknob, might not understand what it is. They may have difficulty remembering spatial locations or spatial relationships among objects. Difficulty finding specific objects against background noise as well. So if an object is indistinct from the background noise, they may have difficulty picking it up. And then finally, one I thought it was difficulty appreciating, interpreting movement of objects in real time. So any moving stimulus may be more difficult for them to interpret than a stationary object. Okay, so I'd just like to acknowledge my colleagues who helped on all these studies, and particularly the patients that we see at the UCSD Shiley Marcos Alzheimer's Disease Research Center. So thank you.